Ladies and gentlemen, please welcome Carnegie Science President, Dr. John Mulcahy.
Good evening, everybody. As you just heard, I'm John Mulcahy. I'm the president of Carnegie Science, and it's exciting to see so many people here and welcome you tonight. For those of you who don't know about Carnegie Science, we're an independent research institution that was founded by Andrew Carnegie in 1902 here in Washington, D.C. And we study all things from very small scales of biology to the Earth and planetary systems to the universe itself. Lately, I've been liking to say we do the origin and evolution of everything, basically, in the universe. That's kind of describes what we do at Carnegie. We are an institution dedicated to basic research, and the need for basic research has never been more important, I think, than this moment in time. One of the other important things we do at Carnegie is public outreach, and I would argue that this is a particular moment in time when explaining to the public why science is important has also never been more important. And so events like tonight are really critical for that. So we really love to see people from the public coming out to hear about science and spreading the word of science. And hopefully you're here tonight, so I assume you all know why science is so valuable. So tonight we're gonna hear about the Kavli Prize lecture series. This is a lecture series we've been doing for a while. And I wanna thank the Kavli Foundation, the Norwegian Academy of Sciences and Letters, and the Norwegian Ministry of Education and Research for their partnership on this series. It's been a really wonderful series. I want to also thank Frank Sesno, who's going to be our host again tonight. You've been doing these for a few years now, so this has been really wonderful. And I want to welcome our speaker, Dr. Kanwisher, who's going to, I think, impress us all with her wisdom about the brain. I guess we're going to hear. This should be very exciting. If you don't know a lot about Carnegie and you want to know more about us, please do. I would encourage you to sign up, go to our website, carnegiescience.edu, to learn more about us. Now, before I turn it over to our speakers, we're going to hear two video messages from people who couldn't be here tonight. The first of these is from Cynthia Friend, who's the CEO and president of the Kavli Foundation. And then we're going to hear from Anneli Eriksson, who is the president of the Norwegian Academy of Sciences and Letters. And we're just going to play these back to back, and then I'll come back up.
Good evening and welcome. I'm Cynthia Friend, president and CEO of the Kavli Foundation. The event this evening, Face Value, How the Brain Shapes Human Connection, will feature 2024 Kavli Laureate in Neuroscience, Professor Nancy Kanwisher, the Walter Rosenbluth Professor of Cognitive Neuroscience at MIT. I regret that I'm not able to join you in person tonight, but I wanted to take this opportunity to share my gratitude for the collaboration between the Carnegie Institute for Science and the Kavli Foundation, which has been a platform for sharing the work and the lives of selected Kavli laureates since the inception of the prize in 2008. The conversation tonight takes on special significance because it's the last in this series. We at the Kavli Foundation are deeply appreciative for having had the opportunity to work closely with the Carnegie Institute for so many years, an organization that shares our strong support of basic science. Now, more about Professor Kanwisher, who is honored for her work that uncovered mechanisms of human visual perception and cognition. Dr. Kanwisher and her lab members asked the question, what is the nature of the human mind? Nancy uses brain imaging techniques to search for regions of the brain that are selectively engaged in perception of faces, places, and bodies, and other regions that are tuned to uniquely human functions, such as music and language. Absolutely fascinating. Dr. Kanwisher is a highly recognized scientist who's known also as an exceptional mentor to early career scientists, a particularly important role today. She's committed to teaching and to reaching lay audiences to share the wonder of human cognitive neuroscience. She's so committed to sharing knowledge and teaching that she famously shaved her head for her video and had one of her graduate students draw the structure patterns of the brain directly on her scalp. And I will say that is dedication to teaching. Perhaps we'll hear more about that in tonight's conversation. The conversation will be moderated by Frank Sesno. And if you've regularly attended these events, you know that Frank has ably moderated many of the Kavli Carnegie discussions. Frank is the executive director of the George Washington University Alliance for a Sustainable Future and founder of Planet Forward. He's a former CNN Washington bureau chief and an Emmy Award-winning journalist. In closing, I want to thank the Carnegie Institute of Science leadership and their staff for their partnership. And thank Frank for helping us make this important science exciting and accessible. Next, Professor Annelin Eriksson, President of the Norwegian Academy of Sciences and Letters, one of the Kavli Prize partners, will share her perspective in a brief video. Our partnership assures the highest level of scientific excellence and integrity in the selection of laureates. So I hope you enjoyed tonight's program, and I thank you for your interest in and support of basic science.
My name is Annelin Eriksson, and I am the president of the Norwegian Academy of Science and Letters in Oslo. I am sorry for not being with you at Carnegie today. It is a great honor for our academy to collaborate with the Kavli Foundation and Carnegie Institution for Science. Professor Nancy Kanwisher at MIT received the Kavli Prize in Neuroscience in 2024, together with Brynrich Freiwald and Doris Tsao, for their discovery of a highly localized and specialized system for representation of faces in human and non-human primate neocortex. Fred Kavli's vision for the Kavli Prize is the recognition of outstanding scientific research in the fields of nanoscience, neuroscience, and astrophysics. To honor highly creative scientists, promote public understanding of scientists and their work, and to foster international cooperation among scientists. This event today is an invaluable contribution to communicate the great achievements of the Kavli Prize laureates. The Kavli Prize was established in 2005 between the Kavli Foundation, the Norwegian Ministry of Education and Research, and the Norwegian Academy of Science and Letters. This collaboration is the key to the international standing of the Kavli Prize. Each of the three Kavli Prizes, astrophysics, nanoscience, and neuroscience, have a prize committee appointed by our academy. The high reputation of the Kavli Prize is due to the solid work by the prize committees. The members of the committees are world-leading scientists within their respective fields, and they are chosen by the Royal Society, the French Academy of Sciences, the Max Planck Society, the National Academy of Sciences in the US, and the Chinese Academy of Sciences. We were delighted to reconnect with Nancy this June at the Federation of European Neuroscience Societies regional meeting in Oslo, where she also took the opportunity to visit our academy. I wish you all the best for today's lecture, Nancy, and I wish you all an inspiring evening. Thank you.
For those messages. All righty, it's now my pleasure to introduce, are we ready? The ambassador, Anniken Huitfeldt, who is Norway's ambassador to the United States since last year. Very new, like me. I started last year too. So come on up and she's going to introduce our speakers. Thank you.
So good evening, ladies and gentlemen. It's such a distinguished honor to be here together with you tonight and gathered to recognize Dr. Nancy Kanwisher and learn from her as a part of the Kavli Prize laureates conversation. As mentioned here, this is a unique cooperation. Along with the Kavli Foundation, the Norwegian Academy of Science and Letters, and the Norwegian Ministry of Education and Research, we are proud of the unique Kavli Prize partnership between government, philanthropy, and academia. Every other year, we recognize outstanding research, promote and celebrate new discoveries, and inspire future generations, encouraging their curiosity and appreciation for science. To signify the importance of this prize, awards are presented by His Majesty King Harald of Norway. And it's truly never been a more exciting year, I would say, to celebrate the Kavli Prize. 2025 is the bicentennial of organized Norwegian emigration to the US. 200 years ago, a brave group of 52 immigrants departed the town of Stavanger at the southwestern coast of Norway, and 53 arrived. Yes, the first Norwegian American was born on that ship, the Restoration. Fred Kavli was born much later, of course, in Ersfjord, Norway, a tiny little village between a beautiful fjord and a beautiful mountain. With igniting his passion for science, like the first immigrants, Fred was drawn to America. Even 100 years later, the US had it was a pull from all over Norway to the US, a land of boundless opportunities and a vast potential. And I mean, the first female professor in Norway, she came to the US to even get a job. She thought that Norway was a dark hole compared to her job and opportunities here in this beautiful land. But Fred, he lived in California. He quickly found success in defense industry. In this work, he see the best of Norway and also the best of America. And he said himself, it was a combination of his Viking courage and his spirit of adventure in his blood. With the Kavli Prize, we celebrate his boundless determination and the enduring contribution to the sciences. And it wouldn't be possible without our tremendous partners. So thank you again to Kavli Foundation for their generosity and sponsorship. Thanks to Carnegie Science for their partnership in arranging this event, and to Martin Luther King Library for housing us tonight. And thank also to you, Dr. Kanwisher, for encouraging our own curiosity and for appreciation for science. So please welcome our presenters to this stage. Thank you.
Fortunately, that didn't happen, and we've actually learned something as a result. No offense. So we gave away the suspense a little bit, but we'd like to invite you, if I can. Okay, this is now not clicking me forward. Can we try that?
Oh, it's there, but it's not on the other
Okay,
side.
but it's
Okay.
not on our monitors here, so, okay.
We gotta look up.
Let's,
There you go.
there we go.
Okay.
All right, there we see it here.
Okay.
So pull out your phones and if you were using the QR code, and we'd like to ask you this, since we're going to talk about brains and recognizing and the places in our brains that recognize faces. How would you say you compare to others at recognizing faces? Much better than almost everyone you know? About like most people? Slower than most? At the back of the pack. Recognizing faces. And we're asking this because there's a distinct place in our brain that helps us do that, right?
Yeah. So we'll see what people say, but my guess is there will be quite a big spread, because in the scientific experiments where people have asked this kind of question, we find there are really, really big differences between people in their ability to recognize faces. We'll see what you guys say.
How do you do?
I'm not so good. And actually, which brings me to another finding, which is that if any of you are feeling like you're not particularly good at face recognition like me, the good news is that face recognition ability is not correlated with IQ. So don't feel too bad.
So if you don't remember a face or a name,
It's
it doesn't
annoying.
mean anything other than.
Annoying, but it's not a general characteristic of your abilities.
Have we got results? Can we put results up? See what we've got? Suspense is killing us here.
It's over there.
Oh, where is
yeah.
it? Oh, there it is.
Okay, so most
Okay.
people were on number 2, looks like about About like most people, okay,
about like most people.
okay.
And we have a lot
Okay.
of smart, we have a lot of pretty quick people in the room too, because there we go. 24% of you say better than most, 43% say about average, 24% again, slower than most, and about 8% of you say, uh-oh, back of the pack. So,
This suggests to me that people are pretty well calibrated, right? There's a range here, which there should be.
okay,
Yep.
so let's get to there we go. Tell us about these specialized ranges.
Okay, so the fact that there's a big difference in people's face recognition ability is just one little sample here. Just take that one little mental ability, and there's a big range, and it's not correlated with your other abilities, like how well you can keep track of where you are when you're navigating, when you're on a hiking trip, wherever you are, or how well you do math, how articulate you are. These things are separate, and we see that echoed in the organization of the brain. These things are computed by different parts of the brain. And so what my lab is focused on for the last few decades, and my broader field, lots of people, not just my lab, is to chart out what these different functions are in the different parts of the brain. And we see that for many parts of the brain, they do very, very specific things. So those little blue dots there that you see, they're on the bottom surface of the brain, mostly in the right hemisphere. And those are regions that we found 20 some years ago. They're present in everyone. We could put any of you in the scanner and find those regions in 5 or 10 minutes of scanning. And those regions respond much more when you look at faces than when you look at anything else.
This is where?
It's about right there. Like if you take your right ear, if you went in about an inch on the bottom surface of the brain.
So how did you find this, and what does it mean?
Okay, so my first year in grad school, the first non-invasive brain imaging study was published on the cover of Science. And I looked at that, it's like, oh my God, that's amazing. Like we can understand the mind, not just by measuring behavior, but by looking directly at activity. And so it took me about 10 years before I managed to claw my way onto a brain imaging machine. And the reason we started looking at faces was that there was already indirect evidence that there might be special machinery in the brain for face recognition. Specifically, people who had damage to the back end of their right hemisphere would sometimes selectively lose their face recognition ability.
Without losing all other abilities.
Exactly. They could still read, recognize objects, get around, no other problems, just only a difficulty recognizing faces.
And when you say measure activity, that's what? Blood flow?
Okay, so the method that I use mostly is functional MRI, and it's the highest resolution method that you can use without opening the head, right? So we can have undergrads come into the lab, pop them in the scanner, and look at activity. But the measure we have is very indirect. With functional MRI, you're looking at changes in blood flow. So just like if you get up and run around, you have to send more blood flow to your muscles because it's metabolically expensive to run for the muscles. When neurons fire in your brain, that's metabolically expensive and the blood flow increases to those parts of the brain.
So what, as we look at this slide here, we see language, speech, music. We talked about faces, we talked about places. These are distinct regions of the brain. What does this tell us about our brain?
Okay, to me, the coolest thing to me is what it tells us about the mind, is that we aren't just kind of generically smart in a very kind of undifferentiated way. Minds are composed of specialized processors that do different things. And so we're specifically smart in particular ways. And so I think that's very interesting. It's been one of the biggest debates in my field for hundreds of years of whether, is a mind like a computer with a powerful CPU, or is it like a computer with a graphics card and a video card and an audio card and all these other bits? Well, we're like the latter thing. We have a lot of specialized bits. And so to me, that's just kind of part of who we are.
And those bits do different things.
Yeah.
Independent from one another?
Well, that's a very good question. They function somewhat independently of each other, but of course, all of these things are also connected to each other and connected to other parts of the brain. And so information must flow around between all of these regions as well. And we know less about that because we don't have great methods to look at it. We do have good methods for saying, okay, how much are the neurons firing here and here and here when you do different things?
I'm going to put up another slide in a minute, but before I do that, I want to ask another question of you. How many of you can recognize a face, but sometimes hesitate or have trouble naming the name.
Like everyone.
Same place in the brain?
No. There's a bunch of dissociable things. Like, in fact, you can already tell the fact that you know who that is. You know exactly who that is. You just can't remember their damn name, right? Like, that tells you those are two different things, right? And in fact, that those blue dots on the bottom of the brain there, that's the part that visually recognizes the face, that kind of matches it and tells you, okay, I know who that is. It's a different part that matches it to a name, and that's further forward in the brain, down the front of the temporal lobe, like a little in front of your ears instead of straight in from your ears. There are other patches that associate with the names.
But they have to speak to one another, essentially, right? Because
Yeah.
first you see the face, you
Yeah.
recognize the face.
That's right, and they
then
go
you
the other
retrieve-
way too. They go the other way too. So we did an experiment way back where we put people on the scanner. We said, close your eyes. Okay, now imagine your mother's face. Now imagine, one after another famous faces. And that same region turns on. Just from the name of the face, if you form a vivid mental image, you can turn that region on. So you can go from the name backwards to those regions as well.
All right, let's do another little experiment here with you. All right, take a look at that. Think for a minute. How many of you can tell what this image is? Raise your hand if you can tell what that image is.
I
Any hands up?
don't see many hands going up.
Can we go to the other one?
Yep.
All right, let's try another. How about that? How many of you can see something in that? Raise your hand.
Takes a moment.
Few hands going up.
All right.
All right. Tell us what's going on.
Okay. So this is the same image. Just on the right, it's right side up, and on the left, it's upside down. So if you look on the image on the right, if you don't have it yet, don't worry, there's nothing wrong with you. It's kind of subtle. It's a profile view of a face. I don't have a laser pointer, but if you look at it, you see there's a dark eye with a nose sticking out to the left. Everybody got that now? Okay. So the weird thing is that the image on the left is the same image, it's just upside down. And so this recapitulates a well-known finding in our field that the face recognition system just doesn't work on upside down faces, right? And so this is actually the first clue that face recognition might be separate in the brain from a PhD thesis actually in my department at MIT in 1969, before any of these methods came along. This guy Robert Inn showed that people are really, really bad at recognizing upside down faces. And they're pretty good at recognizing.
Like in real life?
Yeah. But they're pretty good at recognizing upside down objects and words and other things. And so he said, there's something special about face recognition that it doesn't work when they're upside down. And so that was the first clue that face recognition might be different from other things, just based on that simple observation you all just saw.
So I'm going to keep coming back to this question throughout the evening. So what does this tell us about the region, about the brain, the mind?
Well, okay, what I would say now is that what this tells us is that that region is trained up by our experience, and we usually see faces right side up. And so it's finely tuned to detect differences between one face and another face, so we can tell people apart. But it does that really well with upright faces. It doesn't do it well with inverted faces. And in this case, you can't even tell that's a face when it's upside down.
Right.
And so an interesting sidebar that maybe we'll get to later, but I'll just foreshadow it, is we did some studies with artificial neural networks, just computer programs that are trained with lots and lots of experience to get good at face recognition. These are the reasons why your phone can now identify the photographs. If you don't even ask it and it's sticking your friend's name on their photos, it's like, what is that? Well, that's the advent of artificial neural networks that are suddenly really good at face recognition. And interestingly, artificial neural networks have the same face. It's called a face inversion effect. They're really good with upright faces. They're really bad with inverted faces. And I think for the same reason humans are. They're trained on upright faces.
So interesting. We have a video that's a fascinating video that illustrates some of what's going on with someone who's actually experiencing it. Before I roll that video, though, please explain where this comes from, who this individual is, and how you were fortunate enough to be able to have this.
Yeah. So what you're going to see is a Japanese gentleman who was awaiting neurosurgery at a hospital in Japan a few years ago. And a colleague of mine was working with the neurosurgeon and thought I might be interested. And the reason was that this is surgery before as a treatment for epilepsy. Usually epilepsy is well treated with drugs. If it's not, sometimes the best option is neurosurgery. So then the doctors put electrodes in the patient's brain to map out functions, and sometimes they stimulate to map out functions. And they do that for clinical reasons. We don't go sticking electrodes in people's brains just for the hell of it. We do it only, it's only when a neurosurgeon has a clinical reason to do it. So this neurosurgeon had put electrodes right on top of the patient's face selective regions. And first he mapped them out and showed that those electrodes responded really strongly when the patient looked at faces, not when they looked at other things. But then he decided that he also needed to electrically stimulate that region to really test its function. So the video shows what happens when the guy is stimulated in his face area.
And he agreed to this,
He
obviously.
agreed to this.
Just want to be absolutely clear about that.
And I'm so grateful because he's such a good informant. He's really telling us what he experiences.
It's quite fascinating. So let's just watch this for about a minute and a half, and then we'll come back and
And
talk
he'll
about what
be
it
speaking
means.
Japanese, but there's subtitles.
Right.
And I'll also mention, he doesn't know that there's a face selective region. You can see that in the lower right, he's getting stimulated. He wants to tell us exactly what he's experiencing. I'm so grateful. You can see there he's getting stimulated again. He doesn't know there's a face area. He doesn't know what part of his brain is being stimulated. He's just asked to say what changes. So that shows us that that region is causally involved in face perception. You stimulate there, you change the face percept. We want to know, is it causally involved in perceiving other things that aren't faces?
He's just staring at the box.
Yeah, and he's just asked to report, does any of this change?
秒ぐらい。始まって1秒ぐらいなんか、横になんか目、口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら、気がついたらもうこの箱にしか見えないっていうか。
Do you see a face on the box?
Yeah.
なんすか、またさっきみたいに横に、ちょっと目としてるんで、ちょっと。
Okay, so tell us what,
Okay, so
first of all, how this got done.
yeah, so that's a kanji character and yeah, okay. So what that tells us with brain imaging, you can see activity. That's cool. There's a region that responds more to faces and objects. That's interesting. But you don't really know that it's the region that underlies your face recognition ability until you stimulate it and make a change in face perception. So that's the strongest kind of evidence we can ever get in neuroscience. It's very rare and very precious. I'm so grateful to this guy and the small handful of other neurosurgery patients who have been so generous as to let us learn from their electrodes and their experience.
Was his surgery successful?
Yes, he's a barber and he went back to the barber shop and he's doing fine.
That's great. What does this have to do with sort of the conscious experience, Nancy?
Yeah, well, we already got a hint there, at least with this guy, that when he was stimulated there, it produced a conscious experience of a face, even when he was looking at a box or a ball, right? But that's one patient, one person. We can also get clues in other ways if we look at any of us. Do we want to move on to the glasses, or do you want to look at, yeah?
Okay, you all have glasses on your,
Yeah.
you had
And-
them on your chair.
So I should, if you let me, I'll explain.
Go ahead,
We should
please.
go to the next slide so they can see the red, should be the red, green. Okay, so let me just explain what the glasses are. If you put them on, either way, it doesn't matter. If you close,
Can I take a picture of the audience?
I know,
This is great with it.
this is why I do it, because it's so funny. Okay, so if you close one eye, you should see just a face or just a house. If you close the other eye, you should see the opposite. That's just optics. That's just a way to put a face image into one eye and a house image into the other eye. Now I want you to look with both eyes at the image and just don't do anything, just look. And if anything cool happens, you can go, ooh, ah. What happens?
Shout it out.
Does it switch? Yeah, okay. If it didn't switch for you, don't worry. It's hard to get this to color balance to work for everybody. But I think for most of you, it spontaneously switches between being a face and being a house. Yeah?
How many
Okay.
is it switching for? Is it switching?
Oh, yes.
Look at
good.
that.
It's good. Okay, so we did this same experiment in the scanner because there's not just a face selective region, there's a region that responds to places, including houses. And when we did the same experiment on people in the scanner, we taped those glasses on their faces and put them in the scanner and showed them that image. And they told us with a button press, now I see the face, now I see the house, now I see the face. And the cool thing is nothing is changing on your retina when that happens.
So what's happening in their brains?
So what you see is the face area goes like this, and then the place area goes like that, and they flip every time your conscious experience changes. And that tells us that those regions of the brain are tightly correlated with your conscious experience of what you're seeing, not just what's coming in through your eyes.
Let's just be clear that I just want you to explain something. When you say conscious experience,
Yeah.
what does that mean?
Well, in this case, it just means they tell us what they're seeing. And so you see a face, your face area is active. You see a place, your place area gets active.
They take their glasses off now?
Yes, okay.
Okay. We've got a room full of adults here. They've had a lifetime of recognizing faces and doing things like this. What about, and we were talking earlier, Kathy and I were talking earlier about our grandkids, 2-year-olds. And one of the things I was saying is being a grandparent, which is pretty new to me. Do you have any grandparents in the room? Just the most amazing thing, but it's like an ongoing neuroscience experiment, because you turn around and they're doing something different. It's different than when you're a parent, because you actually have to live with them every day. Here you're dropping in and out, and it's like all of a sudden they're doing different things.
Yeah.
So what about this experience and an infant's brain?
Yeah. So I don't have kids or grandkids, but when I look at those pictures of the brain with all that structure that is essentially the same in every normal person, I think, how do you wire that up? We only have 30,000 genes. How is that a recipe to produce that same structure in every one of our brains? And so I was really lucky to work with my amazing colleague, Rebecca Sachs, starting around a decade ago. And Rebecca worked very, very hard to figure out how to scan infants while they're awake. This is almost impossible. Actually, it's kind of a train wreck. You bring the kid in, the parents are nervous, the kid is squawking. It's just one problem after another. But Rebecca persevered, and actually she went to great lengths to make these experiments work, including producing her own subjects. Her son Arthur was scanned, I don't know, dozens of times in his first year of
I
life.
said, I didn't sign up for this, but he's getting it anyway.
Anyway, the result of that is that we showed that many of these regions are present within the first 4 months of age. And so that doesn't tell us that they're innate in the sense of totally independent of experience, but it tells us that infants are born with a system that's already ready to produce that structure really quickly.
Now, you're able to see a lot of this through the fMRI, right? That you use. Same thing with the children's brains. What are you seeing with an infant? I mean, they're
Well, the images are a lot blurrier because the kids move, and because we can't get as much data, they don't stay in very long. But otherwise, it looks, like a blurry version of what we see in adults. And it's not that it's blurry in their brains; it's just that they're moving and they're not in the scanner as long. So it looks very, a 5-month-old brain, the visual parts, the face and the place areas look very similar at age 5 months and in adulthood.
and it's, we know that this is about the time several months in where they start recognizing themselves in a mirror or maybe their parents. So is this part of a bonding experience?
Much earlier. Kids can recognize faces within a day of birth, which is amazing because their vision is very blurry.
Wait, they're recognizing faces?
They can distinguish their mom from other people within a day. Yeah, there's
Can you measure that?
No, this is behavioral work.
Oh,
So you can ask a newborn infant all kinds of questions before they can speak. And you ask them using a method called habituation of looking time. And that just means it's a way of saying, been there, done that. If a kid has seen something and then you show it to them again, they're bored, they don't look as long. And so you can use that to say, can they tell the difference?
so they're bored with their mom after the first day, basically?
Probably. But so you show them different pictures and
They're recognizing
Yeah.
faces within.
they look, and with parents, they look more at their mom's face than other faces within a day of birth.
And so how does that, what is it? But the region you say that
We don't know. We haven't been able to go down that far. We haven't been able to go down. There's a colleague in Ireland who tells me he's got similar data in 2-month-old infants. But it's really hard to go down to 1 day. What you can use other methods and you can show that some of the same structure is there. Some of the connectivity seems to be present at birth. And so if I had to guess now, nobody knows how brains get wired up, but if I had to guess, because there's still this question, how do all those regions end up in the same place in everybody's brain? How does that work? I think the idea is that the long range connections are all present at birth, and those long range connections kind of instruct what the input is to different regions.
and over time. So let me ask you about a long range connection. Another show of hands, please. If you lost all of your language abilities, God forbid, both
Yeah.
to speak and to understand language, could you still think? You lose all your language abilities, could you still think? Yes, raise your hand. No, raise your hand. Talk to us.
Okay, that's cool. Most of you were right. So my colleague, Ev Fedorenko, has been working for the last decade understanding the brain's language regions. And those have been known for 200 years from studies of patients who had damage to their left hemisphere. But she mapped them out in detail with functional imaging, with functional MRI. And she showed that those language regions are very, very specific just for language. So they do not turn on at all when you do mental arithmetic, when you solve logic problems, when you hold information in memory over time.
You see faces?
When you see faces, they do not turn on at all. They only turn on for language. So that was a real surprise. But even more amazing is work of a colleague of ours in England who's been studying people who have this terrible misfortune of having huge left hemisphere strokes. These are people who lose all language. It's not just that they can't speak, they can't understand language. But what this colleague has shown is that those people can do pretty much everything you would have thought required language. It doesn't. They can do all of those things. They can solve logic problems, they can do arithmetic, they can appreciate
How do we
music.
know that?
Well, she tests them with sophisticated methods where, you can charade, you can gesture, you can show pictures. These people are not stupid, they're perfectly smart. They have simply lost language. And so it shows that all of these other thinking abilities are perfectly intact. And language is just our input-output system that we use to share information with other people. It's very precious, it's very wonderful. It's a key part of what has made humans amazing, is that we build on each other's knowledge by sharing it through language.
We probably all know somebody who's had a stroke and who's had their language affected.
Mhm.
What does this tell us? What does this tell clinicians about what can be done, if anything, to repair that?
Well, two different questions. The first thing it tells us is if somebody's unable to communicate with you, it does not mean they can't think, right? They may well be in there thinking perfectly clearly and just unable to speak clearly about it. That's the really important.
And understand you when you're
Yes,
speaking.
Yes. And so there's kind of some ways to find out. It's tricky, but this is a really important thing. Unfortunately, our field has made less progress in figuring out what to do about it. And so, humans are smart and adaptable. You can't rebuild your language system once it's damaged in adulthood, but you can do all kinds of other intelligent ways to develop workarounds. And so it's a very awful thing to lose your language abilities, but don't give up on those people. Most of them are still there, their same smart selves they ever were. They've just lost language. Yeah.
I'm going to go to another slide in a minute, but I do want to remind you, when you came in and you took down that QR code for Pigeonhole, you can put your questions in there, and I'm going to start weaving in your questions. So feel free to go in there. You can see another question you like. You can tap on it and vote on it, and I'll see what the most popular question is, and I may or may not ask that one. But let's go to this one. And generally, blind people have face areas by touch.
Okay, so this was a study we did a few years ago, this guy Ratan Murthy, who you can see there. And I mentioned before that finding face areas in a 5-month-old infant, that's cool, but doesn't tell us whether it was innate or whether it was developed by experience. So Ratan said, let's scan congenitally blind people who have never seen anything. And so he built these 3D printed objects that you can see there, faces and scenes and hands and chairs. And he put blind people in the scanner and let them feel these objects. And he showed that you get very similar activation, more for faces than for other things, by touch in congenitally blind people who have never seen anything at any time in their lives. And that tells us that those face areas are sitting there in the brain. They're face areas, even if you've never seen faces. I still find this result so astonishing. If it wasn't for my lab, actually, I wouldn't believe it. And the activations are weaker. They're not as strong as a sighted person looking at faces. But look at it, it's recognizably the same place in the brain.
It's incredible. So we've talked about a lot of things, and we'll talk more. And as I say, I'm going to go to these questions in a minute. But you spent your whole life studying our minds, our brains, these places, spaces. I think if we polled the room and asked people, what do you think most about when you think brain? My experience is dementia in my future? Can we do something about that? Will we have a cure to Alzheimer's? Can we prevent this? Will there be a vaccine? From everything you know about our brain, and I know because we've talked about this, you're asked about this a lot. What's your response?
Well, I would love to see positive progress on all of those fronts as much as anyone else. I don't think we're doing great yet. The lore, what we write in our grants is, look, we can't understand how to fix brains or how to predict what will happen to them until we understand how they work in the first place. And I still think that's true, but I do think it's a little frustrating how little progress we've made. Like, we've learned a lot about Alzheimer's and how it develops, but there are not good cures. And even things like depression, we have SSRIs, they're a whole lot better than nothing. Cognitive behavioral therapy, much better than nothing, but not great. There's still lots and lots of people, all of us probably know people who suffer so severely from depression and from other mental disorders. So I give my field a kind of, B- actually in progress so far, but I remain hopeful that this kind of research takes a long time to go from the basic research to the fruits of being able to apply it and make progress.
Which then brings me to this next question that we're going to put to you, right? So into your lab and so many others, and into our lives in so many ways, comes artificial intelligence, this unbelievable thing. So I'd like to ask you what you think. How is the research into the brain regions we've discussed tonight being affected by artificial intelligence? Is it replacing young neuroscientists in the lab? AI can see into the brain and tell us what people are thinking and where. AI is giving you a whole new magnifying glass. AI can predict how strongly these brain regions will respond to a new image. Just tell us more about what's going on there. Or D, AI can't be trusted with this research. So put down your responses and we'll get there. I'm sure you think that AI is a new tool and a game changer in some ways.
It's absolutely game changing for my field. And I say this as somebody who's surprised to hear myself say that.
Why?
Just like 5 years ago, people around me were starting to use AI to try to understand brains. And I was just trying to hide out, waiting for this to blow over, because I don't know any computer science. It's not my skill set. It's not my thing. It just seemed implausible. It's like, that's a computer program. It's not a brain. What are you guys doing? And then I started peeking out of my hole and looking around, and it's pretty amazing. And now half of my lab is training deep neural networks.
All right, let's see if have we got results that we can put up? Okay.
Oh,
Item number
you guys are
3,
good.
AI can predict how strong
You guys are really good. Like, so most people chose C, which is the correct answer.
A bunch, nearly 1 in 5 said it can't be trusted with the research.
Yeah, that's also true. That's also true. Yes.
So tell us about this.
Okay. So yeah, amazingly, we can take an artificial neural network that's just a computer program with a whole bunch of units and a lot of weights, and you train it on millions of examples. This is basically what's underlying the AI revolution. It's why your phone can recognize faces, why it can take dictation, why ChatGPT can predict the next word and give you a sensible answer to a question. All of this is based on massive training with huge numbers of examples on artificial neural networks. And as I'm sure you've all noticed, it's pretty amazing. But the more amazing thing is that actually there are deep similarities between these AI networks, these artificial neural networks, and brains. And this so did not have to be. Like, there's no reason they should have been similar. But if you look in lots of different ways, you see deep similarities. So for example, you can take an artificial neural network that's just trained to recognize objects. You give it an image and it says that's a horse and this is an apple and that's a toaster, right? And it's been trained through millions of examples. Now you take that network and you say, can we match units in that network to responses in parts of the visual cortex in the human brain? And the answer is, oh, yes, you can.
Wow.
I know, it's crazy. I can still hardly believe it. But then, so that tells us, this is really important because before this, we did not have computational models of how the brain does what it does. And now we have a model to work with. It's not identical to a brain, but it captures a lot of what the brain is doing. And better yet, we can experiment on it, right? We can do all of these experiments on our networks. We can do things like control experience. I can't take an infant and say, okay, if they grew up and never saw a face, how would they be at face recognition? That would be unethical. But I can do that with an artificial neural network.
Is this exciting or just purely creepy?
I find it thrilling because I can answer questions I couldn't
No,
answer
I mean, I
before.
get it, but it's like you have this
Okay, let me tell
computer brain, right?
but let me tell you my favorite. There's many cool things we've done, but here's my favorite. I used to end every talk for about 15 years saying, okay, we have all these special parts of the brain. Why is that a good way to design a brain? Why not just have one generic smart brain that can do everything and doesn't have different pieces? I have no idea. I never thought I'd be able to answer that question, but now I think we can answer it. And here's a little hint. One of my postdocs a few years ago took an artificial neural network, and she trained it both on object classification and on face recognition. So she fed it 500 different kinds of objects, toasters and trees and apples and whatever, 500 different categories, and 500 different people with lots of different images each. And she fed it all these images until it got really good at classifying each image, saying either, that's a horse or that's Joe Schmo, right? So it gets really good at it. And so then she looks inside and says, okay, how is it organized? And damned if it hasn't spontaneously segregated itself into two separate systems, one for object recognition and one for face recognition, just like we see in the brain. And to me, that says, this is just what an optimized solution looks like. Brains have been optimized through evolution and learning. This thing has been optimized through machine learning tricks, and they come up with the same solution. They both discover here are two different problems. We'll allocate different parts of our system to solve those problems. Isn't that cool?
I guess so. I mean, I want to go on to other things, but before we leave AI, there's a lot of excitement about AI, and I have some of that. I have very interesting ethical conversations with students about whether they and how they use it.
Yeah.
Fascinating.
Yeah.
But there's also something, and we see it all the time. It's a little science fiction-y, weird, Hal in 2001, creepy, right? This thing's going to do things. And now, where we are now, we're teaching it empathy. And I don't know how many of you have talked to, ChatGPT, the voice, and back and forth. I'm sorry you're feeling that way. That's a very good question. Thank you. It's like, so is it, what do you think of what is going on?
I think large language models are like ChatGPT are extremely interesting. They're very, very empowering for our research. So ChatGPT also captures a lot of what goes on within those language regions in the brain.
Right.
We can get those similarities. That's really interesting. But what I think the language models don't do is, they just get the statistics of language. And I think that's also true of the brain's language system. It's operating on the statistics of language. But to understand language in a deep way, you need to ship that information out to other parts of your brain that can build models of the meanings that you're processing. And I think we can discover a lot of that, of how that might work in the brain by playing with models that just have the ChatGPT part versus other models that also have vision parts and other parts.
Where's the field going?
I think that's a big part of where the field's going, is that now we have these computational models that capture a lot of what's going on. They are not perfect at all. There's a lot they do not capture about what humans do. And actually, interestingly, a lot of the things that the AI models are bad at is just basic vision, like not knowing that that's a glass or a table or a person. They're good at that. But knowing that the glass is resting on the table, knowing that I better not do that. That would not be good, right? Never put everyone on edge. Their AI models are not good at that. They can't tell the glass from the table. They can't understand the physics of the situation. So one of the key things we're working on in my lab now is how humans understand the physics of the world they're looking at, which we need to do to conduct any action on the world. And AI models are not so good at that yet.
I'm going to go to audience questions in just a minute, but before I do that, one thing I do want to ask, two things I want to ask you about. Mentoring. This came up before, that you believe so powerfully in this. How many in the room are in the sciences, were in the sciences? A lot, right? You are working with young scientists and aspiring scientists. You know, it's one of the most amazing things about being in a university. But you were mentored. You had a pretty fierce person. You were ready to bail, and she kind of wouldn't let you.
I had an incredible mentor. I have several of them, but one in particular. Yeah, like, you know, I dropped out of grad school 3 times to go be a journalist. I did.
That's going to be a separate conversation.
Separate thing, but it's true. And I just had this incredible mentor who, you know, after years and years of failed experiments, I don't know what she saw, but she's like, no, you're going to stick with it and you're going to finish and I'm going to stand by you. And she saw some ability in me that I absolutely did not see in myself. And there's no way I would be here now if I had not had Molly Potter, the most amazing mentor ever.
And your mentoring?
Well, I try to pass it on, like, because I think it's actually the most important thing we can do as a scientist, not just out of compassion for young people, but even narrowly, what is the biggest scientific impact you can have? The biggest impact you can have is through the multiplier of your students. Collectively, they will do so much more than you could ever do in your own lab. And so the best impact you can have is to train good scientists. Plus, it's fun.
Well, it's an incredible and very personal investment in the future, which leads to my next question about investing in science.
Yeah.
And the funding for science.
Yeah.
You said to me that your own lab might not survive.
Yeah.
How worried are you about it?
I'm very worried. My lab only has funding for the next 1.5 years. And I only have that funding because I've been at MIT for 25 years and I've been saving discretionary funds all that time.
Your funding was coming from where?
Mostly NIH. But, you know, that we are in serious trouble in this country right now. Like the NIH budget, $2 billion was thrown out of this year's NIH budget. Poof, right? 1 billion was thrown out of the NSF budget this year. And the administration's request for the science budget next year is a 36% cut in non-defense research. These are drastic cuts in science. I'm not taking graduate students now. They apply to my lab and I say, I do not have funding. I can't take you.
So where are they going?
Right. Hopefully there's other labs who have better funding, but most of my colleagues also don't have funding. It's serious. They're going to other countries. You know, we have been the global leader in science for nearly 100 years, and we are flushing it down the toilet right now.
I don't want this is not a political question at all.
It's just a fact about what's happening.
It's a science question.
Yeah.
What will happen to science if these funds are not in some way from some place?
I think it's going to take over a generation to recover if we ever restore funding. It's not like a tap. You turn it off and you turn it on. Right? If you're in the middle of a study, a longitudinal study, like some of the pediatric cancer studies that were just halted, imagine that. Childhood cancer, we're no longer funding. Cut it off, right? You can't just start it again. You know, those study subjects are, you know, who knows where they are. You've fired all your people. You can't go rehire them. They're doing something else now. So the long-term effects of cutting off science are severe and they're going to be very long-lasting.
Well, let's hope this.
I'm sorry to be the bearer of this awful news, but I think it's very, very
Well,
serious.
it's important that people hear it, and it's important that the efforts to change some of this continue, because maybe
Yep.
some of that can be
The
effective.
sooner the funding gets restored, the greater the chance that it could be rebuilt.
Let's come to some questions from you all here. We see more faces on our phones than real ones some days. Is there such a thing as face overload? And can this impact our ability to recognize faces in real life?
That's interesting. I think we use the same machinery for face recognition when we look at a screen and when we look at an actual face. Actually, I didn't do this study, but a colleague of mine did this heroic study where they actually asked, does it matter if it's a real face or a photograph? Usually we just use photographs in the scanner because it's too hard to get a whole bunch of people to march by. But she got a whole bunch of people to march by the scanner and peer in the tube. And it was pretty similar. So I think it's similar machinery we use in our brains when we recognize.
The message is just put your phone down.
Yeah, right.
Here's one that plays right out of what we were just talking about from Theo. How can current and aspiring scientists push back and survive in a society that refuses to fund research without leaving it?
Yeah. I don't know. I worry most about young scientists, grad students, postdocs who are just, trying to launch their careers right now. I think it's a really, really tough time, and I don't know what the advice is other than to make the case and make it clear that this is not something we can just shut off and wait and it will recover. That's not going to happen. And so I think we just need to share this message with everybody, because I think people don't realize what the huge costs are.
I was at a science conference at the French embassy just very recently, and they made a big thing of their campaign, Choose France for Science. They're trying to attract American scientists to France.
It's trying,
Right.
I mean.
So is that where our students are going to go?
Well, France, and I'm going to Norway if I can't. I'm serious. I am. If I can't run my lab, I have a plan. Trondheim, the Kavli Institute. That's where I'm going. All of my, not all, a lot of my science colleagues are plotting where they will have to go because we are all worried that we cannot run our labs. It's just all. And I will say that, one of the original things that American science was built on was in the 30s was the exodus of Jewish scientists from Germany. They built American science, right? So German brain drain was a major original impetus for American science. Well, now maybe we're going to feed science around the world, which is.
I hope that doesn't happen. Here's one. Has research been done with other species, such as monkeys and chimpanzees, that spend more time upside down on their ability to better recognize faces upside down?
That's a great question.
I love that question.
It's a great question, but I'm going to start by saying, yes, there's loads of research with monkeys, and actually all the limitations we have with studying humans, because we can only rarely, when a neurosurgeon sticks electrodes in, we're very excited, but we don't stick them in ourselves. That's unethical. But with monkey research, they've been able to go far beyond what we've seen with humans. And monkeys also have face areas. There are lots of beautiful studies. In fact, the Doris Tsao and Vinra Feiwald, with whom I shared I was going to call it the Kavli Prize. They did that research in monkeys and showed with great precision the same face areas in monkeys, going much farther than we can do in humans. But to get to the question, there are studies of whether monkeys have face inversion effects. And I think monkeys do have smaller face inversion effects than humans do, probably because, as the questioner said, they spend more time upside down.
So they can recognize their fellow monkeys upside down. Okay, that's good to know. The initial slide showed brain regions active related to other people's thoughts. Is this related? Great question. To empathy.
Yeah. So that I didn't get to mention it before, so I'm glad that the questioner asked. That was the work of my colleague Rebecca Sachs, the same one who did the infant studies, and she showed amazingly the little patch of the brain right there gets active whenever you think about what someone else is thinking. And, she was my grad student in the early 2000s. And when she first started finding this, I'm like, Rebecca, that's charming, but I don't believe it for a moment. Go back, do more control experiments. And she did dozens of control experiments. She's an amazing scientist. And it's just true. Every one of us has a little patch of brain that is dedicated only to thinking about what other people are thinking. And if that's
Some of us have a larger patch than others.
Well, wouldn't you think? Weirdly, actually, okay, I scanned my partner and he doesn't have it. He claims,
How you doing?
well, one could think that's incriminating of him. I think it's actually more incriminating of me. But actually, I think he was just trying to second guess and rethink the design. And I don't know.
Is he
Anyway,
an empathetic person?
he's a very empathetic person, so I can't really explain it. But he didn't have that patch. Anyway, most people have it. I'm pretty sure he has it too.
Here's another very interesting question. Do you know what happens to the brain when someone's in a coma?
Oh, it's a great question. It's very hard to tell when somebody's in a coma. Are they in there, right? And this is really important for loved ones to try to figure out, are they still there, right? Excuse me. Luckily, there's been some incredible research over the last 15 years. I don't have anything to do with it. I just watch. Where they've been able to show some of these people can't move, right? If they're locked in, they can't control movement, and so they can't speak. They can't tell you what they're thinking. But what this guy did was to put them in scanners and ask them to think different kinds of thoughts, kind of like the imagery experiment I described. Imagine you're walking around a house. Imagine the appearance of a face, right? And he showed that when they gave them that instruction, the coma patients turned on different parts of their brains. And that means they understood what they were being told. They were able to actively turn on these different parts of the brain. They're very much in there. This is not true for all coma patients, but this research has been going on long enough that the ones who can do that, it turns out, are the ones who are more likely to come out of the coma later. So that's
Is that
pretty
because there are
important.
different levels of coma or different
Yeah.
types of coma?
Yeah. And some comas are people who just have lots of brain damage and they're not going to come back. But some are people whose brains are actually mostly intact. They just got stuck in this unconscious state. Yeah.
How many people in the room have a pet? Okay, this next question from the audience is for you. Is it the face or the object region of the brain that recognizes faces on animals, cats, dogs, horses?
Face region, done those experiments. Yeah. Early on, we showed pictures of cat faces, and the face area responded as strongly to cat faces as human faces. And at first, I was kind of appalled. It's like, what? But cat faces are pretty compelling. There's a reason we like cats and relate to them, and I think they activate the same regions.
Well, that's encouraging. Here's another very interesting one. Using music as an example. Is there a difference in what reaction you're able to measure depending on the different genre, I guess, of music that's played for a subject?
I'm sure there's a difference in the brain because we can tell the difference. And it's not happening in the toes, it's happening in the brain. But that's really hard to tell so far. What we do know is that there are populations of neurons in auditory cortex that respond much more strongly to music than to any other sound. Actually, we do know a little teeny piece of that. We can tell the difference between vocal music and instrumental music. In fact, there are populations of neurons that respond only to vocal music. They don't respond to instrumental music, and they don't respond to speech, which we didn't anticipate at all. It's pretty wild. But we see that in a lot of subjects.
One of the most fascinating things that I've seen is, these, and I'm sure you've seen it too, these remarkable experiences of patients with pretty advanced Alzheimer's who can remember us. They'll actually sit down and sing or play a song. It's the most astonishing thing.
Yeah.
What's happening in the brain for that?
Well, I think
And is that same
I think
region?
it's consistent with this picture that I've been painting, that the brain isn't one homogeneous thing. It has these different parts. And if your music system, or also your motor cortex, it's controlling how you play, those memories are still in those parts of the brain. So that person may not recognize a family member, certainly may not know their name, but their music regions, their stored motor music production are intact. And this is kind of what you'd expect from this modular organization of the brain that I've been talking about.
Stay with that for just a second. So my mother-in-law lived to 100.
Wow.
She was amazing. And she was playing tennis into her 90s.
Wow.
But in the last couple of years, she started to experience dementia. And she got to a point where there seemed some recognition when someone walked in the room, a family member, but she didn't connect it with a name. So in a situation like that, what's happening to that region? What do we know about that region and how that's functioning when a brain is deteriorating like that?
Yeah. what do we know about that? Well, I haven't done those experiments and I'm not totally up on that literature, but I think with something like Alzheimer's, it starts in certain parts of the brain and then it kind of spreads around and probably it starts in the middle of the temporal lobes and spreads around. And you get thinning of the cortex, which is where a lot of the processing happens. So there's just a lot less of the machinery is still there. And so you still get some of the activation, people retain some rudimentary abilities even when they're pretty advanced in Alzheimer's, but there's just a lot less of the machinery is there.
Here's one related perhaps to this. What's the primary obstacle in repairing the language centers of the brain once they are damaged?
Yeah. I think that nobody knows, but my guess is that the reason, well, first I'll say, if there's damage to language cortex in childhood, there can be a lot of recovery. Those regions can move around. If the damage happens before puberty, there's a lot of plasticity in the brain. But if the damage happens in adulthood, it's a much more serious problem. They can't move around so much. And I think part of the constraint is that the connections between different brain regions are already fixed, and those can't really shift around. And so you can't just train up a different piece of cortex to do language for you because it doesn't have the right inputs and outputs. That's my guess.
I have to say, we have some really remarkable and actually some very personal questions here. We have a few more minutes. I want to go through that and then we'll move on. But here's one. My daughter is autistic. Given the vast difference in how autistics perceive and process stimuli, how much of all you've discussed tonight differs from holistic to autistic?
Yeah, it's a great question. I spent several years trying to study autism. I found it really difficult. And the main finding, we were studying very high functioning autistic people because that's who will go in a scanner, right? And so our main finding is that all of the things I've talked about tonight were exactly the same in the people with autism, including, remarkably, that region that's selectively responsive when you think about other people's thoughts, which is sort of the core function that people tend to think of as a central challenge in people with autism. That region was still there, the same size, the same location, the same everything, which is just very surprising. Although I will say my colleague, Rebecca Sachs, the amazing Rebecca Sachs, did a study where she looked not just at the existence of that region, but what kind of information it encoded. And in typical subjects, if you look at the pattern of response across the little pixels in that part of the brain, she could decode in a typically developing person whether they were thinking about someone else's action as intentional or accidental. That made different patterns in there that she could decode. And this matters, for example, the difference between murder and manslaughter. It's the same action. They just differ by intention. So these are things we care about. Our legal system cares about. They matter. And that difference is encoded in that part of the brain. But she could not decode that difference in the people with autism. But that's a very subtle difference. All the same regions were there. And to me, that's just a statement about how weak our methods are. Because obviously, if you have a person with autism who struggles a lot with certain cognitive and other challenges, something's different in the brain. And if we can't really see it with our methods, it's a statement about the methods. We need better methods in our field.
Yes. Here's related. Schizophrenia has been recently linked to the ability to see. Does that mean that it is posted, says this question in quotes, in highly specific areas of the brain and can thus be targeted?
It's a great question. And a colleague and I were just given a grant to start studying schizophrenia. We're just at step one. I know almost nothing about it, but we've just started sticking our toes in that literature to try to see what's done. There's lots and lots of studies. I'm not going to say much because I just haven't learned much about it. I don't think there are any clear messages that this particular part of the brain is affected. People have been focusing a lot in neuroimaging studies of schizophrenia on the frontal lobes because these are kind of higher order reasoning areas that if you'd had to guess, schizophrenia is famously a thought disorder. The frontal lobes are the first place you'd look. But there don't seem to be really clear, straightforward findings yet. And certainly in any of these cases, you can't tell from brain scans whether a person is completely typical or autistic or schizophrenic or anything else, which again is just a statement that our methods are not good enough to see things that are clearly different in there.
A follow on this, I guess. I recently learned about the potential for telepathic communications between children with non-speaking autism. Can you talk about recognition and language?
I don't know anything about that. I mean, I do think that, again, we humans are very, very good at reading each other's thoughts, right? We have this special brain region that does that. Even people with autism who develop, to be reasonably functioning, they do gain those abilities. They use that same part of the brain when they do it. I think this is a kind of core human ability to understand each other's thoughts. And it's there in rudimentary forms in very young infants, although the full-fledged ability takes a while to develop. So I don't know about this particular finding in kids with autism, but I think it's a core human ability.
Last question from the audience. I'm going to give you an opportunity to reassure everybody in the room, because we had, says this questioner. What if we didn't see the image switching between a house and a face?
Don't worry,
Is
it's
that-
fine.
Is that
It's
because
fine.
I'm watching on a computer, or do I need a doctor visit?
No, you don't need a doctor visit. People have different ratios of red and green receptors in their retinae. And these glasses they're not even exactly the right color for that image. So it's close enough that it works for most of you, but it just means you have a slightly different ratio of red and green receptors in your retina. And that's nothing to worry about. There's a big variation across people in that.
Nancy, your science is amazing, and what you've done and discovered is remarkable. But before we let you go, part of the objective of these conversations with Kavli and Carnegie is not just to learn about the science, but the scientist.
Mhm.
So tell us a little bit about, what do you do to relax? What do you do to not be thinking about the brain all the time? What are your hobbies?
Well, I'm a pretty narrow-minded scientist. I really do hang out in my science cave most of the time because it's fun and I love it, and it's just what I want to be doing. But I do occasionally do other things. My current fixation is I'm trying to plan an adventure that may or may not happen, but I'm trying to put it together for January. I like hiking and kayaking and outdoor adventures, and I like the kind that have a little bit of innovation involved. And so I have two inflatable sea kayaks. They each fold into a 35-pound duffel bag. And my partner and I have taken them to very remote parts of the earth and taken them out and pumped them up and just gone off for a week. And right now I'm trying to plan a trip in a part of Indonesia called Raja Ampat, which has the highest marine biodiversity in the world with beautiful coral reefs. And so the plan is to fly with our kayaks and duffel bags to Raja Ampat, pump them up, and go off for 10 days. We'll see if I pull that together.
And I hope you'll send pictures and tell us more. But you kayak on the Cape.
Yeah, I won't admit how many kayaks I have.
Go ahead.
Kind of embarrassed.
Go ahead.
8.
She collects kayak. Any kayakers in the room? Yeah, okay.
Yes, so I grew up on Cape Cod in Woods Hole, which is a lovely place where pretty much everybody's a scientist. So, I'm not one of these inspirational stories of somebody who clawed their way to science against all odds. No, my dad was a biologist, and I published my first paper with my dad, and I was sneaking into the science lectures when I was in high school and all of that because it was all around me. But it's also a good place to kayak. We have all these islands and passages and fun places to go. So, yeah.
And I'm going to let you end by telling us when you shaved your head and had your student draw. And I was listening to one interview, you were referred to as a badass scientist.
Well, a long time ago, I thought, it's like a lot of my colleagues write books, and I always felt like, oh God, what's the matter with me? I haven't written a book. And it's like, it's just writing is a struggle. So I thought, well, I give decent lectures. Maybe I'll just put some of my lectures on the web. So I put a bunch of lectures on the web, on a website, and nobody knew about it. So it's like, how do I draw eyeballs? It's like, oh, I need to make a viral video. And right around then, I was diagnosed with lymphoma. And I had to undergo chemotherapy, and it was clear I was going to lose all my hair, and that was, bracing. But I also thought, oh, what an opportunity. So I hired a film crew, and I started a lecture, and I said, well, where are all these things on the head? You can't see it with this hair in the way. let's deal with that. And I just shaved off my hair with the film. Actually, I had a fantastic camera person, and he made me practice. Like there's this part where I snip off a big chunk of hair. And he said, that's only going to read if you get the angle right. And made me practice it like 15 times until I got the angle right. Anyway, it's kind of ludicrous, but it's on the web. You can go look at it.
I
Anyway.
thought you have quite a map.
Oh,
in
yeah.
your style.
And then, okay, so then I shaved my head, and then we wanted realistic brain anatomy. So my grad student used actually maps we had made of my brain with an external system for figuring out where they are, which I won't explain, it's complicated. And she drew on my head anatomically correct neuroanatomy, and it was pretty crazy.
And your hair grew back and you were.
It did.
This is a
I'm totally cured and I'm fine. Again, thanks to basic research.
Well, I want to thank you for both, obviously for a fascinating conversation, but for the incredible work that you have done and for the advocate that you are for science and the funding of science, for the mentoring you do for next generations. They will figure this out. They may have to claw and fight and scratch their way to do it, but they will figure this out. I'm quite confident of that. So
Awesome.
Thank you very much.
Thank you so much. It's been super fun.
All righty. Thank you. Thank you, Nancy and Frank, for that really interesting conversation. And thank you for those great questions. That was really some remarkable questions. I really enjoyed that. And I want to once again thank the Kavli Foundation, the Norwegian Academy of Science and Letters, and the Norwegian Embassy here in the US for their support of the event tonight. And if you want to know more about Carnegie Science, I would encourage you to visit our website, carnegiescience.edu, or you can find us on Instagram, Blue Sky, or Facebook. And we hope to see you at another event soon. Thanks, everybody. Let's thank our speakers again.